Okay, well, thank you, Will. So Will just talked about how he used a method called target trial emulation to evaluate vaccine effectiveness, but one of the really great things about having access to a large amount of these routine data, linked data in OpenSAFELY is that we have the flexibility to approach research questions in different but complementary ways and respond quickly to current public health questions. So today I'll be talking about back in autumn 2022, the NHS offered a COVID booster along with the flu vaccine. So initially they offered it to people who were considered clinically vulnerable and over age 65. But then in October, they expanded the eligibility to people aged 50 years or older who were not clinically vulnerable. And this decision was made on the best available evidence at the time and to mitigate a probable winter COVID wave. So there's ample evidence that COVID vaccines are effective, and boosters are also effective as well when they're received. But the question of the effectiveness of vaccination campaigns, it's highly dependent on the context in which they're implemented, and so it's really important to understand the impact that these campaigns are having in order to better plan future campaigns. So this plot here, this shows COVID booster coverage by 1 year of age, and this was done in nearly 2 million people. So these are non-clinically vulnerable people between the ages of 45 and 54, and these are primary care patients, which we identified in OpenSAFELY. And as you expect, we see a rapid increase in the coverage rates after the booster vaccine first became available, and it plateaued after about 6 weeks. And of course, this is exactly what we'd expect. But the reason this is interesting is that we know at the population level that 49-year-olds and 50-year-olds, they're extremely similar in terms of their health status and their clinical characteristics. Because nothing magical happens when you turn 50. You're almost exactly the same person you were when you were 49.9 years of age. But because of the way this particular public health intervention, that this vaccination campaign was implemented, they do differ in one very important regard, and that's in terms of their vaccination coverage rates. And we refer to this as a natural experiment. So the provision of an intervention, in this case the booster vaccine, based on an age-based threshold, this is a natural experiment, like I mentioned, because treatment is not based on individual factors. It's not based on the patient's health status. It's not based on the prescriber's preference. It's just whether or not they're above or below the age of 50. And so this mimics randomization, this process. It results in minimal confounding, and we can take advantage of this. If we use the right methods and we apply them properly, we can actually get causal estimates of vaccination campaigns and the vaccines themselves. So the thing is, these natural experiments, they're occurring all the time when new policy interventions are introduced. So not just vaccination campaigns, all sorts of things, new drugs are introduced to the market, that sort of thing. And we're very well placed within OpenSAFELY to evaluate these sorts of interventions in a timely manner. So I think I want you to consider this particular talk a case example of how we can take advantage of these natural experiments to evaluate public health interventions. And so in this particular case, in order to evaluate this particular natural experiment, we use something called regression discontinuity analysis. And one of the really nice things about this sort of study design is actually answer two types of questions. First of all, what is the effect of the actual campaign itself on health outcomes? But we can also look at, well, what's the actual impact of receiving the vaccine on health outcomes? And for those of you who are more statistically minded, we use something here called instrumental variable analysis. But just in the interest of time, I'm really only going to focus on the first question here. So in this plot, this shows uptake of the vaccine. This is 6 weeks after the campaign had been rolled out. And we can see that vaccine, so even though 100% of people over 50 were eligible, the coverage rates were quite modest, with under 50% of people actually getting the vaccine. But again, as we'd expect, we do see a large discontinuity in those vaccine coverage rates between 49-year-olds and 50-year-olds. So the question is, do we see a similar discontinuity in the outcome rates at that threshold of 50 years? Because if we do, then there's a good chance it's attributable to the vaccination campaign itself. So we focused on 3 outcomes. First of all, we looked at severe COVID outcomes, that's hospitalization, A&E visits, and mortality. We couldn't look at things like COVID testing because at this stage in the pandemic, COVID testing is not very well captured. We looked at respiratory admissions and mortality, and lastly, we looked at all-cause unplanned admissions as well. And a couple of things I want you to take away from these plots. First of all, if you look at the y-axis, first of all, these outcomes are all very rare, which is a good thing, probably reflecting the fact that there's a relatively healthy and younger population. But again, the other thing we can see in these plots is we do not see any discontinuity or any drop in the rates of the outcome at age 50 like we did in the vaccination coverage rates in this plot here. And if we analyze these data, this is what we get. I'm going to talk you through this. But these point estimates and their confidence intervals, these represent the estimated change in the outcome rates associated with the vaccination campaign. And we had, the reason there's multiple dates there, we looked at a range of 6-week periods. We had a pre-vaccination campaign period, and this is our negative control because we wouldn't expect to see a change. And we also looked at a range of different time periods throughout the vaccination campaign because we know that vaccines do take a little bit of time to become effective. So we want to see if it changed over the course of the winter. And so what we can see here is that there was no observable effect of this vaccination campaign on any of these severe outcomes. And why is this? Does this mean that the vaccine is not effective? Well, no, because we have lots of evidence that they are. But this is very much about this particular individual vaccination campaign, and these results may not necessarily hold for future campaigns. So there's a couple of reasons why this is what we found. First of all, in this population, the vaccine coverage rates were quite low. So as I mentioned, it's about 40 to 50%. The COVID infection rate in the population at the time, it was very low. Only about 2 to 3% of people in this age group became infected during this time period. There was quite high pre-existing immunity in this population as well. We know at this time and point, based on other data, that nearly everyone had been infected with COVID. 85% of this population had 3 or more vaccinations already, so the marginal benefit of an additional booster was probably quite low. And lastly, again, being a very relatively healthy, very youngish population, the outcome rate is very low, so which, again, is a good thing. Like I mentioned, we couldn't evaluate the impact on infection rates. We didn't look at things like, how does this impact people taking time off work, sick notes, and obviously there's also a benefit to vaccinating younger people, like herd immunity, having an indirect impact on those who are clinically vulnerable. And I'll just note that since this analysis, the WHO has actually updated their guidance. They no longer routinely recommend boosters, vaccines to young, healthy adults, and the NHS does similar as well. And just lastly, I just want to make a case for using more of these natural experiments and taking advantage of them. It's sometimes an overlooked study design, but again, we're very well placed in OpenSAFELY to take advantage of them more than we are doing currently. And if you're interested in this topic, I encourage you to check out our work because there's a lot more detail about the methods, about different results and sensitivity analyses that we've done as well.